Hello everyone, this presentation will focus on microcystin production on the millennial scale. It is a paleolimnological investigation of two subtropical lakes in central Florida, USA. Harmful algal blooms have been increasing in modern lake ecosystems worldwide. While cyanobacteria play a key role in nutrient cycling and have shaped the Earth's history through the Great Oxygenation event, their overgrowth and increased production of cyanotoxins in recent decades threaten ecosystem services, biodiversity, and public health. Among the cyanotoxins produced, microcystins are the most cosmopolitan. Among many drivers, their production has been linked to elevated phosphorus and nitrogen levels, stoichiometric ratios, high cyanobacteria biomass, and zooplankton grazing. However, the ecological function of microcystins and the precise environmental triggers remain poorly understood. The increasing frequency of harmful algal blooms is often linked to cultural eutrophication due to human-driven nutrient loading. However, few paleolimnological studies focus on the long-term history of cyanotoxins, and our current knowledge is limited to water monitoring over the past decades. Our objectives in this study were to identify the Holocene trends of microcystins productions in two subtropical lakes, to link the stratigraphy of microcystins with changes in nutrient inputs and primary producer composition, and to investigate the role of past climate in shaping the patterns of microcystin deposition. The study focuses on two hypertrophic lakes in central Florida, Lake Dora and Lake Marion. Both lakes frequently experience harmful algal blooms with cyanotoxins. Lake Dora is heavily influenced by human activity. Since 1887, the construction of the Apopka-Beauclerc Canal indirectly connected Lake Dora with Lake Apopka, which is known for being nutrient polluted. Lake Marion, in contrast, is more agriculture dominated. Since the 1880s, a network of canals was constructed to drain wetlands and reduce flooding in the area. It significantly altered the lake's hydrology and lowered the water levels. The Florida Department of Environmental Protection has taken monthly microcystin measurements from Lake Marion over at least the past 2 years, and then shown in the picture, microcystins were detected almost in every month. The toxicity of most of these measurements exceeded the EPA's limit for drinking water, and in some instances approached the limit for recreation use. So we performed fieldwork and collected sediment cores from both lakes. The sediments were analyzed using various laboratory techniques. We analyzed the samples for loss of ignition, bulk density, carbon, nitrogen, and different elements such as phosphorus, iron, and sulfur. We performed pigment analysis, which is a useful paleolimnological tool because it enables to reconstruct primary producer communities over thousands of years. Cyanotoxin analysis was performed using ELISA kits to measure total microcystins in the sediments. We also analyzed pollen in Lake Dora and charcoal in Lake Marion to serve as proxies for past climate conditions. The sediments were dated using lead-210 and radiocarbon dating of macrocharcoal, and these dates were fed into our beacon to produce age-depth models. Microcystin concentrations were divided into 3 zones of low, moderate, and high microcystins using hierarchical clustering, and both lakes showed a consistent chronological pattern. Moderate microcystin levels were detected in the bottom sediment zone, while the highest levels were observed in the top zone. However, the chronological spans of the microcystin zones varied between the lakes. In Lake Dora, moderate microcystin levels occurred under dynamic nutrient conditions with increased abundance of photosynthetic pigments. The High pollen counts of Mars taxa indicate a more extensive wetland environment during the modern microcystin interval. Low macrocystins were linked to stable nutrient conditions, the lowest total phosphorus, high total nitrogen to total phosphorus ratios, and relatively lower pigment abundance. An increase in pollen taxa indicative of wetland forest occurred around 2.5 thousand years before present, and it accompanied a very gradual increase in total phosphorus, but it didn't trigger any notable changes in primary producers or cyanotoxins. The high microcystin levels were associated with a rapid rise in most of the nutrients, the highest total phosphorus levels, and low nitrogen to phosphorus ratios, as well as an increase in cyanobacteria, more specifically nitrogen fixing cyanobacteria. The paleolimnological record of Lake Marion exhibited a more complex paleolimnology than that of Lake Dora. There were signs of water lake level fluctuations, which may have prolonged moderate microcystin concentrations. The aquatic environment from 7.2 to 6,000 years before present was characterized by the presence of cytonimin, which is a photoprotective pigment found in benthic colonial cyanobacteria. Cytonimin can indicate lower lake levels where the damaging UV radiation can reach the benthos. During the same period, the charcoal record indicates a higher fire activity, possibly due to drier conditions. Increased fire activity after 3,000 years before present had no synchronous shifts in microcystins, and the transition to from moderate to low microcystins at 2.3 thousand years before present was associated with relatively more stable nutrient conditions characterized by the lowest total phosphorus levels, high nitrogen to phosphorus ratios, and relatively low pigment abundance. Similar to LDO, Lake Dora, the high microcystin levels were associated with increases in nutrients and primary producers abundance, as well as nitrogen fixing cyanobacteria. So both Lake Dora and Lake Marion exhibited elevated cyanobacteria abundance and moderate microcystin levels during their early limnological stages. This was followed by a prolonged period of low cyanotoxin production until the relatively abrupt transition to very high microcystin levels in the most recent centuries. We attribute this sharp increase primarily to phosphorus loading, which enhanced eutrophication and created stressful environmental conditions that could trigger the production of microcystins. While phosphorus plays a dominant role, we acknowledge that the post-depositional transformations of nitrogen may mask the historical importance of nitrogen in the observed microcystin concentrations. While due to cultural eutrophication, the most extreme microcystin levels are found in the top sediments, it was feasible to reconstruct microcystins over millennial timescales. However, to fully understand how cyanotoxins are preserved or degraded in the sediments, we need more paleolimnological research. In this study, we investigated the sedimentary record of total microcystins and other paleolimnological variables in two hypertrophic lakes in Florida over the past 7,000 years. We found that microcystin concentration in the sediments were associated with total phosphorus and other primary producer pigment concentrations. This shows that apart from cyanobacteria, microcystins are triggered by the increased presence of other primary producers, suggesting that the broader aquatic community is involved in cyanotoxin ecology. Periods of ecological stability where pigment and nutrient levels remain relatively unchanged, had the lowest microcystin deposition. Although hydroclimatic variability influenced nutrient inputs over time, proxies like pollen and charcoal showed no direct connection with microcystin deposition, suggesting that climate was a secondary driver of cyanotoxins. In contrast, the highest microcystin concentration occurred during the last few centuries when hypertrophic conditions developed due to anthropogenic impacts. These impacts included landscape alterations and hydrological changes that led to accelerated phosphorus deposition and intensified cyanobacterial dominance. Apart from the authors of this presentation, I would like to thank the Corning team for making this research possible. You are welcome to contact me if you have any further questions, and thank you for your attention.